Good afternoon, everybody joining us from various places across the world and predominantly from the Philippines. Thank you so much to Mark for this invitation to address you guys on some of the underground killers, in other words, major soil-borne diseases of potatoes. I'm Jackie van der Walt from the University of Pretoria, Department of Plant and Soil Sciences. So potatoes have no lack of diseases. They have a plethora of diseases. So for a plant pathologist, it's really fun to work on potatoes. Not so much fun to be a farmer working or growing potatoes. In this presentation, I'm going to discuss four main groups of diseases that affect potatoes. And these are powdery scab, common scab, rhizoctonia diseases, and then soft rot and blackleg. So let's start with powdery scab. Powdery scab and root galling are caused by Spongospora subterranea, FSP subterranea, and throughout the presentation I'll either refer to it as SSS or Spongospora just to make it easier. So some of the diseases caused by Spongospora are really increasing in incidence, severity, and distribution globally, and this we've seen over the past decade to 15 years. Some of the reasons for this increase are that Spongospora diseases, in other words predominantly powdery scab, have been underestimated and misdiagnosed. It's quite easy to confuse common and powdery scab, even to the trained eye. In South Africa particularly, and perhaps in other parts of the world too, there's been a stigma related to the disease. It's now dissipating, but previously growers with powdery scab in their fields did not want to admit it. So for many years it went undetected. Another reason for the increase in this disease is that the spores are able to survive for a very long time in the soil, and there are very few chemical or other kinds of controls that can actually eradicate these spores from the soil. And then the intensification of potato production globally has resulted in an increase in this disease. There's a limit to the amount of virgin soil available. Another problem is that many of the cultivars that we plant are actually highly susceptible to this pathogen, which also results in an increase in disease. And then in South Africa particularly, the irrigation of the crop also exacerbates the problem. So what diseases are actually caused by SSS on potato? Well, the first one is a root infection caused by zoospores, and this results in disrupted root function. Now, this is not that easy to see in the field. You need a microscope, but this is definitely one of the diseases caused by Spongospora. The second is root hyperplasia, which results in root galls, and these are visible to the naked eye, and these also result in disrupted root function. The third disease, and that's the one that we normally associate with Spongospora, is powdery scab. And this is a tuber blemish disease, but it does result in reduced quality of the tubers and in rejection of seed batches. So let's start looking at the life cycle of this disease so that we can understand when it infects the potato and where the critical points of control are. So the primary inoculum source is resting spores which are found in Cystisorhi in the soil or in seed, on seed. And this primary inoculum, as I've said, can survive in the soil for 20 to 50 years. Anecdotal evidence from Scotland says up to 50 years in the soil without a host. The other problem is that it can survive passage through the digestive tract of animals, which some other spores can't do. So the two primary inoculum sources, as I mentioned briefly, are contaminated soil and infected seeds. So this is where we need to focus our attention in terms of management. So why are these Cystisorhi so highly resistant to all sorts of control strategies? Well, let's have a closer look at the Cystisorhi and what they're made up of. When viewed under an SEM, the Cystisorhis looks like a grain of sand. And you'll see that there are many individual components as part of the Cystisorhis. And each of these little balls, if you want to, is a single resting spore. Now, each Cystisorhis can contain between 700 and 2,000 resting spores. And inside each resting spore, there is a zoospore. Now, when a plant starts releasing root exudates, it will then trigger the zoospores inside the resting spores to germinate, to swim towards the roots, and to subsequently infect them. And this can be as quick as 15 minutes. Now, the problem is that firstly, the resting spores have 3 layers of cell walls, which makes them highly recalcitrant. And the other problem is that when the root exudates trigger zoospore release, not all of the resting spores within one cystisorus will release their zoospores. So this means that even if you have 10% of the zoospores being released, you have somewhere between 70 and 200 zoospores being released at a single event. And bear in mind that there are thousands, millions, actually trillions of cystosori in a contaminated soil. So if you do the maths, you realize that there are millions of zoospores being released in a single event that can infect plant roots. So how can we reduce primary inoculum? Well, first of all, choice of field is very important. If possible, choose pathogen-free fields with no history of Spongospora. If this is not possible, there are other options, which I'm going to get to in a moment. Fields should always be well drained. The pathogen swims, so therefore it requires water, and if the fields are not well drained, you're going to create a favorable environment for the pathogen. Planting pathogen-free seed is also important. Obviously, confirming that seeds are free from the pathogen is not a very easy thing to do, but the best one can do is to plant certified seed to ensure that the level of pathogen on the seed is as low as possible. Some biological control products have been shown to have some promise in managing this pathogen, and these include those with Trichoderma or Bacillus as active ingredients. Various soil and seed treatments have been tested over the years, and none of them have been shown to be 100% effective in controlling Spongospora. Some that have shown a level of control include propaneb, formaldehyde, mancozeb, fluazinum, flusulfamide, metamsodium, calcium cyanamide, and then zinc oxide and certain sulfur and copper containing compounds. But these are only effective when you have a very low pressure in the soil. Moving on from primary inoculum to secondary inoculum. The secondary inoculum is in the form of zoospores, and these, as I mentioned, can swim, so they move in soil water to infect more plants. So this is where we're going to start looking at the critical points in the disease cycle. So the critical tuber infection period occurs just after tuber initiation, bearing in mind that different cultivars initiate at different times, and some cultivars will initiate for a long period of time. For the development of powdery scab, infection of tuber initials must occur by 40 days after emergence. If infection does not occur in that 40-day period, then you're unlikely to see the development of powdery scab. And if infection occurs really early in the season, then one will notice more powdery scab than when it occurs later on in the season. Critical temperatures for tuber infection are between 9 and 17 degrees Celsius soil temperature. The other critical points are the infection of root hairs. Infection of root hairs, in contrast to that of tubers, occurs throughout the plant's life cycle. It starts around 15 to 20 days after the roots are formed, and it will then continue until the roots die off. So zoosporangia and root galling can be observed from 15 to 45 and 45 to 75 days after inoculation, respectively. So you'll see that within 2 weeks after inoculation, one can start observing root galls on roots. Critical temperatures here are between 11 and 25 degrees Celsius. So summarizing some of the important temperatures for infection of potatoes by Spongospora, the minimum temperature, these are soil temperatures, is around 9 degrees, optimal between 12 and 16, and maximum about 25 degrees. So what's important to note is that one should choose a planting date such that the temperatures during the first root formation are greater or higher than 20 degrees C. In the life cycle of Spongospora, we have two different kinds of life cycles. We have a complete life cycle and an incomplete life cycle. In the complete life cycle, which occurs in primary hosts such as potatoes and various others that have been discovered more recently, cystosori, or the resting spores inside the cystosori, are produced again, and this subsequently increases the soil inoculum. So one wants to avoid any kind of host in which the complete life cycle of Spongospora can be completed in a rotation cycle with potatoes. On the other hand, in the life cycle, we also have an incomplete life cycle where the pathogen is unable to produce cystosori after infection of this plant. So it does not increase the soil inoculum. And these are the kinds of plants that one would want to include in a rotation cycle with potatoes. These would either be non-hosts or what we call trapping crops. Now, in some studies that we've done, and we've subsequently done more such studies, which would hopefully be published soon, we had a look at some of the common crops that are planted in rotation with potatoes in South Africa to have a look at whether they are non-hosts, hosts, or trap crops of Spongospora. And this gave us some alarming results in that a lot of the crops that are planted very commonly in rotation with potatoes, for example, wheat and maize, are actually hosts to Spongospora. So what I did was I had a look at Spongospora hosts and asked myself, but what about other pathogens in the soil? So if a grower is trying to make a choice about which crop to plant, what are his options? So most growers will know what the major pathogens are in their fields, and in that way they can make a decision on the rotation crops that are wisest to plant. So already we can see that some crops should probably not be planted in rotation with potatoes if you have problems with Spongospora, Streptomyces, and or Rhizoctonia. And these include onion, cabbage, carrot, radish, and tomato. So we're not left with very many options. So which just makes it more challenging to be a potato grower. To get back to Spongospora, how do we ensure that we have healthy plants? Well, we need to avoid over-irrigation and waterlogging, particularly during tuber initiation, to ensure that the pathogen cannot swim to its host. The levels of plant nutrition, particularly of nitrogen, need to be monitored carefully, and high levels of nitrogen must be avoided. Cultivar choice is important when we're dealing with Spongospora. Now, although there aren't any truly resistant cultivars to Spongospora, there is a range of susceptibility. And what has been noted in studies that we've done and various other researchers is that there is no relationship between root and tuber susceptibility. So one cannot assume that if a cultivar produces root galls, it's necessarily going to develop into powdery scab or vice versa. Now, these differences are differences between research studies and also between the root and tuber susceptibility are due firstly to environmental conditions, because there's a very strong gene by environment interaction, different agricultural practices that affect the susceptibility of the cultivar, the pathogen distribution in the field, the time of tuber initiation, planting date, and the pathogen population in the field. Nevertheless, it's always wise to try and plant a cultivar that has a higher level of tolerance rather than one that is highly susceptible. Right, we're now going to move on to common scab. Common scab is often confused with powdery scab, but it's a completely different pathogen that causes common scab. The pathogen that causes common scab falls into the genus Streptomyces, and there are various species that are responsible for causing common scab. These are actinomycetes. They are basically gram-positive bacteria, although they grow like a fungus. They are filamentous, and they're very widely spread in the soil. Some of the Streptomyces species that are known to be pathogens on potatoes are the following. The most common one, and the one that we know causes common scab on most potatoes, is Streptomyces scabiei. And other species include Turgidis scabiei, Europaea scabiei, and Ascites scabies. The pathogen range is increasing with more and more studies that are done. So this is not an exhaustive list of the pathogens that are responsible for common scab. So what are the symptoms that we note with common scab? The initial symptoms are small, brown, raised spots on tubers, which can enlarge, they may coalesce, and eventually they become dry and corky. And this is where the big difference is. With powdery scab, the lesions are, as the name suggests, powdery on the inside, whereas with common scab, the lesions are normally corky. They're tan to brown, they're normally roughly circular or star-shaped, and they can extend quite deep into the tuber depending on the pathogen, the environmental conditions, the cultivar, and the interaction between these factors. So there are three basic common scab lesion types, and I'm going to move to the next slide to explain them. The first is a netted or russet scab, which is very superficial, but it could look a bit like a russet tuber variety. The second is superficial scab, and this is what we often call just normal common scab, star-shaped lesions which don't go too deep into the tuber. And then lastly is deep-pitted scab, where the lesions will actually go more than 5 millimeters into the flesh of the tuber. Right, moving on to the disease cycle of Streptomyces. The initial inoculum comes from infected seed tubers, firstly, or from spores or mycelium that survive in the soil or on plant debris. So, as with powdery scab, we have seed as an initial inoculum source as well as soil. We need to remember that infested soil can be disseminated between fields by wind or by rain, and infection occurs during tuber initiation. Again, bearing in mind that tuber initiation takes place at different times and can be shorter or longer in different cultivars. What we need to bear in mind with common scab is that high soil moisture will reduce common scab. High soil moisture during tuber initiation will reduce the incidence and severity of common scab. So this is in contrast to powdery scab. The other thing we need to remember is that common scab is common in neutral or alkaline soils. So in other words, if one acidifies the soil, one will generally see a decrease in the amount of common scab. So the Streptomyces pathogen is actually adapted to feed off the dead or corky tissue that the tuber produces in response to infection. And this is why we see these corky lesions. It's actually the tuber's response to infection by this pathogen. So the more the tuber responds with producing cork, the more the pathogen is able to infect. Again, this pathogen has a fairly wide host range, particularly on root or storage organs, and these include radish, turnip, beetroot, groundnut, carrot, parsnip. So these should be avoided in rotation with potatoes. Looking at management, there are very few treatments that are 100% effective. Seed treatments is questionable because a lot of Streptomyces species are found in the soil, and soil treatments often give inconsistent control. Irrigation management is one of the best ways to actually reduce tuber infection, and this should ensure that the soils are moist, not waterlogged, but moist during tuber initiation. And again, this is only if one doesn't have a severe problem with Spongospora. Green manure is partially effective. use of clean or certified seed, and then again long rotations with non-hosts of Streptomyces. Right, we're now moving on to Rhizoctonia diseases. So the symptoms of Rhizoctonia are quite diverse. The ones that we normally or have previously always associated with Rhizoctonia solani on potatoes is, for example, black scurf, which I'm showing you here, stem and stolon canker here and here. Okay, however, in recent years, a number of other symptoms have also been associated with Rhizoctonia, and I will discuss some of these in this presentation. So Rhizoctonia solani is a basidiomycete fungus, and it's responsible for disease in numerous plant hosts. It has what we call 13 different anastomosis groups in this species complex, and these AGs differ from one another. This is why it's important that we understand this complex. They differ in terms of their pathogenicity, virulence, host range, fungicide sensitivity, and symptoms caused. So it's very important that growers and others involved in advising growers know which Rhizoctonia AGs are present in their soil to be able to control them properly. So why do we need to know which AGs are present? Well, we know firstly that AG3PT is most predominant in potatoes, and this is the AG that develops sclerotia, which causes black scurf on potato. And these are the survival structures of this AG. We also know that the previous crop will affect the AG prevalence in the field. So for example, if soybean was previously planted, you might find a prevalence of AG4HG3. With maize, you might find AG223B, and sugar beet AG4 or AG22. What's important to note here is that AG4HG3, such as on soybeans, or 223B, will infect potatoes under warmer conditions than AG3PT might do. So where we normally associate cool and wet conditions with infection by Rhizoctonia, this is not necessarily the case for all of the AGs. It's also important to note that the AGs differ with respect to their sensitivity to fungicides, and we published a paper not so long ago on this. So one cannot use a blanket fungicide to treat all the different or to control all the different AGs found in a field. And they cause different symptoms, and this is important to note. So in general, Rhizoctonia solani AG3PT prefers cool temperatures between 5 and 25, high soil moisture, organic matter, and a pH of less than 7. But as I've mentioned, this is predominantly for AG3PT. So some of the other symptoms that are caused by different AGs are, for example, stem and stolon canker. Various AGs cause this. Aerial tubers. Aerial tubers are often an indication of few or no marketable tubers to be found underground. So this is quite something to take note of. Black scurf, the most common and obvious sign of Rhizoctonia. And then cracked or malformed tubers, which are caused by different AG groups to AG3PT. So looking at the Rhizoctonia solani disease cycle, we will again start with initial inoculum. The primary inoculum sources are contaminated soil and seed. Again, same as with Spongospora and Streptomyces. So the pathogen will overwinter as Sclerotia and Mycelia in the soil and on plant debris, and it is also introduced to the soil or to the field through infected seed. So how do we manage Rhizoctonia? Well, again, pathogen-free soil and seed where possible. Shallow planting is important because we need to ensure that the potato plant emerges as quickly as possible so that it does not, so that stolon canker and damping off do not develop. Avoid waterlogging Same as with Streptomyces, we do not want the potato plant to have wet feet. Plant nutrition is important, and in this case we want to increase phosphorus, sulfur, manganese and copper to increase the plant's resistance to Rhizoctonia. We want to increase nitrate applications and decrease ammonium applications because alkaline soils will suppress Rhizoctonia solani. Soil and seed treatments. No fungicides have been found to be 100% effective in controlling Rhizoctonia, and what one also needs to remember is that resistance develops in the Rhizoctonia solani population and in different AGs at different rates. Various effective biological control options are available, and most of them focus on using Trichoderma species to manage Rhizoctonia. It's important to harvest early, as the disease will develop after vine kill and again in storage. So get the tubers out of the soil quickly to prevent additional development of sclerotia on the tubers in the soil. Hygiene is very important. It's important to disinfect equipment and stores to prevent further spread of the pathogen and also development of disease. And then crop rotation, again, same advice as previously, a 3 to 4 year rotation with non-hosts, for example, grains. And one should actually develop region-specific rotation programs depending on the specific AGs found in an area. But let's move on to soft rot and blackleg. This is a disease complex, so we're dealing with a number of different symptoms and different pathogens. So the disease complex includes the following symptoms: soft rot, as you can see in the middle photo, blackleg, the top two photos, and some of the others down the bottom, aerial stem rot, lenticel hard rot, and non-emergence as in the bottom right photo. So the pathogens that cause this disease complex are grouped as the soft rotting Pectobacteriaceae. They used to all be in the genus Erwinia, but were subsequently reclassified and placed into Pectobacterium and Dicea genera. Now the four species that I have highlighted there, Pectobacterium atrosepticum, Pectobacterium brasiliense, Dicea solani and Dicea dianthicola, are the ones that cause most of the blackleg problems worldwide. Dicea solani is highly virulent, as are atrosepticum and brasiliense. So these are the ones that we normally focus most research on. What's very important to remember about all of these pathogens is that they are bacteria, they are facultative anaerobes. In other words, low oxygen levels or high carbon dioxide levels are favorable for their growth. And this is created through moisture on plants or on tubers. So we need to avoid layers of moisture on the plants or on the tubers. Also important to note is that these bacteria can only penetrate plant tissue through wounds or natural openings. As with Spongospora, these bacteria have flagella and therefore they're able to swim. So immediately we know that water is very important for disease development and pathogen dispersal. Now, in contrast to all the other diseases that we've discussed today, in this case, the primary source of inoculum is seed, not soil. The bacteria are primarily seed-borne and they cannot survive in the soil for extended periods of time. So the conditions that are favorable for disease development are high soil moisture, soil temperatures above 20 degrees C, dense canopies, because that again creates a high moisture around the plants and the tubers, and then wounds and other stress conditions. So when we're dealing with a seed tuber, if a small or a very thin layer of water surrounds the seed tuber, it can rot very quickly, because tubers can become anaerobic when a 30 micron layer of water is around the tubers for either 6 hours at 10 degrees or 2.5 hours at 21 degrees C. Obviously anything in between as well. So it's important to note that if a tuber has a very, very thin layer of water around it, it's going to respire anaerobically, which is going to create a favorable environment for the bacteria to multiply and to cause tuber rotting. So looking at the disease cycle of soft rot and blackleg, we know that seed is of primary importance. So once the bacteria are in the field, they've arrived through seed, we also find that they can enter the fields through contaminated irrigation water, through insects, not as true vectors, but as indirect vectors, transmitting the bacteria on their body parts. They can also be transferred or disseminated through nematodes and aerosols from neighboring fields that are contaminated. What's very important to note is that the bacterial load in the vascular tissues, the lenticels and the wounds of seed tubers or of harvested tubers will determine the risk of blackleg or soft rot respectively. So this is what we want to minimize. We want to minimize this bacterial load in tubers. It's been found in literature that the minimum threshold value for disease to develop is approximately 10 to the 3 colony forming units, in other words 10 to the 3 bacteria per tuber, which is extremely low. So approximately 1,000 bacteria per tuber is the threshold value for disease to develop. So control strategies aim to minimize bacterial loading in seed tubers, because the higher the starting inoculum level, the sooner rotting will start. However, control is highly erratic depending on the prevailing environmental conditions. So this disease and the development thereof, and also the rate of development, is extremely dependent on the prevailing environmental conditions. So what do we do to manage this pathogen? So there are some strategies that can be applied pre-plant and at planting. So pre-plant, one should use early generation seed to ensure that the numbers of bacteria in the seed are low. Clean the packhouse regularly with disinfectants such as chlorine. Apply fungicides to seed. Although the fungicide is not going to affect the bacteria, it's going to hopefully kill off fungi that might create wounds, which would allow infection points for the bacteria. If seed is cut, ensure that the cutting equipment is regularly cleaned and disinfected. And in so doing, ensure that the cutting wounds have dried and suberized before planting these seed tubers. Tubers should be planted in dry conditions when the temperatures are below 20 degrees C. And tubers should never be planted when they are wet, because, as I've mentioned, a wet tuber is an anaerobic tuber. Physiologically mature tubers should be planted so that, as in the case of Rhizoctonia, so that they can emerge and start photosynthesizing as quickly as possible. Some cultural practices that can be followed to minimize this pathogen, or to minimize the disease rather. Remember that because this is a bacterium, there are no registered chemicals for control of this disease complex once the pathogen has entered the plant tissue. There are some biocontrol products which show potential, such as phage therapy. I know that in the UK, some phages have been commercialized for use against soft rot and blackleg. Ensure that potato plants receive adequate calcium and magnesium fertilization to strengthen their defenses to this pathogen. Reduce nitrogen applications. If the plant receives too much nitrogen, it's going to grow vegetatively, which will essentially make it what we call a soft plant, allowing for easier infection by the bacteria. Again, a minimum of a 3-year crop rotation program. Control weeds and volunteer crops, because these can play a role as hosts of the pathogen between successive potato crops. Ensure good drainage in the field, as with Rhizoctonia and Spongospora management. Rogue infected plants where possible, particularly in the case of seed crops. And do not pulverize the haulm during wet conditions. If one pulverizes the haulm, it's likely that you're going to spread the bacteria, particularly in wet conditions, from diseased to healthy plants. When dealing with harvest, there are some other practices that should be followed. All machinery, not only harvesting machinery, should be washed and disinfected regularly. As with planting, harvest early in dry, cool conditions. Don't allow tubers to lie in the sun, then they're going to rot faster. Minimize tuber damage as far as possible, so harvest gently. If possible, particularly this is easy if harvesting by hand, remove rotting and mother tubers before or at harvest so that they do not land in the same bin as the healthy tubers. And then place tubers at room temperature before transport to prevent condensation. So cool the tubers down gradually. So in storage, as I've mentioned, the tubers should be cooled slowly and in a very well ventilated area to prevent any condensation around the tubers. Seed should be cured properly before placing in storage. And as far as possible, remove any damaged or rotten tubers and do not place these in storage with healthy tubers. Adequate ventilation, extremely important. If there are pockets where there's poor ventilation, one is probably going to find pockets of rotting. Again, I cannot emphasize this enough, clean stores, bins, graders regularly with a disinfectant. However, it's important to first remove organic matter with something like a high pressure sprayer and then apply the disinfectant. If tubers are washed for whatever reason, ensure that wash water is changed frequently and treat the wash water with disinfectant. Ensure the tubers are dried properly after washing. So that's a summary of Pictobacterium and Dickia diseases and how to manage them. If we pull all these diseases together and we have a look at the golden thread running through all of them, we can see that it all comes back to the disease triangle, actually the disease pyramid, that plant pathologists rely on to manage diseases. First of all, we need to ensure that the let's start with the environment is conducive for plant growth and suboptimal for pathogens. So one needs to understand the disease cycle and the conditions under which disease develops and try and avoid those favorable conditions for disease development. We need to make sure that the plant, the potato in this case, is as healthy as possible and strong to launch its resistance mechanisms against the pathogen. And then, as far as possible, reduce inoculum, reduce the spread of inoculum, and just basically reduce sources of inoculum in and around the potato plant. So with that, I would like to conclude, and I would like to acknowledge numbers of students over the years. This is by no means an exhaustive list of all the people that I need to acknowledge. Funding for most of these projects has come from Potatoes South Africa and the National Research Foundation of South Africa. And thank you so much for your time. And now I'm sure that the floor is open for questions.